to repeat that. Okay, so for our case study of promisces, it's related to a pilot-scale evaluation of advanced technologies for the removal of PFAS during landfill leachate treatment. As we said, we are focused on on the matrix of landfill leachate. We are trying to develop an innovative approach to treat the landfill leachate by enabling safe resource recovery later, and then provide technical also background information of the opportunity to use the reverse osmosis and nanofiltration to treat this type of matrix. We should be taking into account, obviously, the PFAS and toxic removal and also the water valorization if possible, and also technical and economical aspects, and also some LCA and LCC analysis to check the feasibility of this type of needs. To reach the design of pilot, we started first by conducting a mass balance of PFAS in a full scale, let's say landfill leachate. We selected 3 landfill leachate are in Italy. We did the sampling campaign to check the fate and the pathways that PFAS are taking during the normal landfill leachate or conventional landfill leachate treatment plants. We did a mass balance for the 3 plants, and we were able to see clearly from the mass balance that in conventional leachate landfill treatment plants, let's say, the PFAS goes and exits as it is. So we can see that there is no removal, more or less, except if we can see in general mass balance, which we had an RO, which is the resources at the end of the plant. So the permeate was in a very good condition. The only problem that also the concentrate in this case was treated later and sent back to the wastewater plants. In any of the cases, so this PFAS is not removed, it's only getting moved from one spot to another, and this problem is huge. There you can see a quick comparison of the 3, let's say, full-scale landfill leachate mass balance, and as I said, we have no removal, no treatment at all, more or less. So, because the main idea that this kind of conventional landfill leachate treatment were not built to remove PFAS in the first place. That's why we need an advanced solution. So one of the solutions that was suggested is the removal first, which is to test the filtration system, nanofiltration, reverse osmosis, the possibilities of this kind of technologies, the removal of PFAS. We tested 2 kinds of configuration, one without recirculation, so 1 step going, and the other one is without with recirculation, sorry, around 50%. To check also the possibility of stressing, will it affect the removal, the fouling, checking all this data parameter on the effectiveness of the suggested technology. We checked nanofiltration around 20 bars, and the other was around 50 bars of pressure. You can see here clearly in this picture the pilot hole that was selected. We put the pilot hole in an actual full-scale, let's say, treatment plant. We are taking the effluent from the effluent of the landfill leachate treatment plant to treat it and check. And you can see also on the right an inside picture of the hole, which you can find also. We put specific sensors that are obviously done to monitor, let's say, monitor online some kind of parameters, temperature, pH, the pressure, etc., and the flow, obviously. Here you can see a quick table of some parameters that we selected during, let's say, the normal runoff, which is, let's say, we had permeate flow. We normalized all this for 5 cubic meters just to understand how much we are having. You can see that we had a recovery without recycling nanofiltration about 34% of the flow as a permeate, and in the RO about 24%. This obviously increased when we had a recycle, but also with it increased the consumption. You can see in this slide just a quick overview of how the flow obviously will be reduced over time due to the fouling that is happening inside the, this was inside the, let's say, the filter of the membrane. This was done only with normal operation without recycling, just to check how much fouling we have, when we need to rewash our system, etc. You can see here a comparison that we did without and with the circulation of around 36, 37 days of working. You can see a great difference with fouling when you have the circulation, which also expected. But we wanted to check if we stress out our membrane too much, will it affect the removal? Will it be still in good efficiency? And how much, let's say, when we circulate more, how much time we need to wash? So all this data also will be needed later when we do our LCA analysis and LCC costing. As for the results of the PFAS, we can see we tested obviously 30 types of PFAS measured. You can see written in small lines which were not detected at all. The ones that were detected are reported in this table. You can see that it's all reported as in nanogram per liter. You can see that in RO permeate there was nothing detected, so RO was perfectly removing. At least it was below the detection limit. Which is 29 gram per liter. And for the NF, let's say, permeate, it had also a good efficiency in removing this type of PFAS. We reached a very, very good results, which is below the limits also that set. There is no specific limits nowadays for the PFAS, but still, if it's still below the limits that are suggested nowadays. For the rejection, you can see a good rejection overall for the PFAS, which is almost reached 100% for the RO because we did not detect, and for the nanofiltration, almost around 93%. But still, after we treated the PFAS, let's say removed actually the PFAS, we moved this kind of pollutant from the influent to the concentrate. So we did not let's say destroy the PFAS. We just only moved it from one matrix to the another. So one of the suggested solution was to try to treat the concentrate that we are producing in our membrane. And this one of the treatment that we suggested was that co-releases of the concentrate that is produced from the membranes with the sludge of the landfill leachate. Because in the conventional landfill leachate treatment, there is some sludge that is produced and also sent to, let's say, later to landfills again or to treat in some thermal way. One of the options was to try to treat them together to have some resource recovery and also to save the efficiency in maybe removal or destroy for the PFAS. So you can see here the prototype, how it looked like. We had a feeding of landfill leachate and the concentrate, and we had three products, syngas, biochar, and bio-oil. For, as you saw, that the concentrate that's going from the nanofiltration and reverse osmosis is actually a liquid. So we need first to try to evaporate it. So use then a normal evaporator just to concentrate as much as possible to try to dry the concentrate, to make it dry material like salts. So we see that here we have also condensate. We check the condensate for the probable, let's say, movement of PFAS because during evaporation, because we reach evaporation at 110 or 140 degrees in that evaporator, we need to check if there is some kind of movement with the evaporation to the condensate of the PFAS. But after analysis, there was no detection, which means that all the PFAS were concentrated in the dry material. Then we did some pyrolysis test, as we mentioned, for the landfill leachate, landfill sludge actually, plus without and also plus the concentrate to check the possibility of what is the pathway that is happening with the PFAS where it goes. We did as we said, we are producing biochar, bio-oil, and we did the analysis for all of them. You can see in the table there is this analysis of the raw leachate sludge and the RO dry concentrate. You can see a very high concentration of PFAS. But on the other hand, after let's say trying to analyze the kind of biochar and bio-oil we had, there was no PFAS detected in the bio-oil at all. And the biochar, only 2 samples, we found some BFA and BFPS, but were in very, very low concentration. So in other, let's say in other words, the PFAS were not detected in the biochar or in the bio-oil at all. So if we just go back a little bit here, the only let's say one exit that we still need to check, we are now working on it, it's syngas. Because the only, let's say, product that we have, we need to check if there is some kind of PFAS in the syngas that are still in the evaporated with the gas that's going out. We did some experiments for the syngas to check also the possibility of valorizing this kind of product, the possibility of the use. You can see that from this kind of quick analysis that there is almost a good percentage of methane, which this means that it can be used as a energy source later. Obviously, there is some CO because it's without oxygen, this kind of treatments. But still, nowadays we are doing the checking for the PFAS inside this kind of syngas. For the last step now, after getting all this material, all this data, this all data will be later used for the SEA analysis to check, because after now we do our, we have all our bank, let's say, bank data of this treatment from the removal to the destruction in the treatment in the bioreleases. We need to check the possibility of the usage of the upscaling also of these kind of technologies by doing an NCA and NCC. You can see here clearly the boundaries and the scenario that has been defined. Now we had finished the data collection. We made some literature review to compare with other similar, let's say, work similar to this one. But now we only need to do this CNCC and also finish with the SINDAS to check the possibility of upscaling our treatments. That was it for me. Thank you for attention. And if you have any questions.
I have one question here.
Yes.
Thank you for your presentation. I have a question on pyrolysis.
Yes.
What is the temperature profile?
Okay, so we are now testing different temperatures reaching from 400 degrees to 600 degrees. And in all the temperatures that we tested, there was no PFAS detected in the biol and biochar. So in different temperatures, we check to see if there is a, let's say, maybe at lower temperatures, we will have more PFAS, but still not detected.
The question is, I know that carbon regeneration
yes.
is done at higher temperatures.
Okay.
Why did you choose?
Because the idea.
Above 800.
Okay, but the idea was not to produce activated carbon. This was not the objective of this kind of treatment. Because in the idea in the first place, the landfill leachate originally as a sludge and also as a RO concentrate will have a high contamination, not only PFAS, but also other types of contamination. So obviously it will not be used later to, let's say, for any, as a soil amendment or as a, let's say, as a carbon. The idea, yes, but the idea was to try to first see that is it a possibility to remove some kind of this kind of pollutant, this one. Also try to decrease the amount of, let's say, what will, because, the sludge that will be treated, eventually the sludge after the finish, the end line of the sludge will go into landfill again. So the idea also to decrease this amount that we have, but it was not an objective to create an activated carbon. That's why
this was not, sorry, it was not what I